Hello, my dear friends. Welcome. Now we are going to continue the chapter 11 Transport in Plants. Okay, so let's continue plant water relations. Water is essential for all physiological activities of the plant, not only the plants, whatever the living organisms, most around 70% of the body is made up of water. Because of that, water is very important for physiological activities, nothing but for the functional activities, and plays a very important role in all living organisms. It provides the medium in which most substances are dissolved. Because of that, we call the water as universal solvent. The protoplasm of the cells is nothing but water in which different molecules are dissolved and several particles are suspended. A watermelon has over 92% of water. So most herbaceous plants have only about 10 to 15% of fresh weight as dry matter. So remaining around 85% will be water. Of course, distribution of water within the plant will vary. Like not all the organs, not all the parts will have an equal percentage of water. For example, for woody parts have relatively very less water, while the soft parts mostly contains water. A seed may appear dry, but it still has water. Otherwise, it would not be alive and respiring. Terrestrial plants means the plants on the land take up a huge amount of water daily, but most of it is lost through the air through evaporation from the leaves. That is, through transpiration. The process of evaporation of the water from the leaves, we call it as transpiration. So, a mature corn plant absorbs almost 3 liters of water in a day. Because of that, the corns will be very fleshy and soft in nature. While a mustard plant absorbs about equal of its own weight in about 5 hours. So there it requires, if they are planting the mustard crop, it requires more amount of water. Because of this high demand for water, it is not surprising that water is often the limiting factor for plant growth and productivity in both agricultural and natural environments. Because of that, the water plays a very key role in agriculture. Next, water potential. To comprehend the plant water relationship, an understanding of certain standard terms is necessary. It is very important concept. First is water potential. So, water potential, psi W, is a concept fundamental to understanding of water movement. So, what is potential? Potential means the energy present in the water. It's like we have solute potential and pressure potential. Solute potential is psi S and pressure potential is psi P are the two main concepts that determine the water potential. So it is very important we have to know about solute potential and pressure potential. Solute potential is nothing but the potential, the energy which is possessed by the solute, and pressure means itself will have some pressure. Water molecules possess kinetic energy, means the energy which gains for the movement, we call it as kinetic energy. In liquid and gaseous form, there are in random motions that is both rapid and constant. The greater the concentration of water in the system, greater is the kinetic energy. For example, we have two substances in which in one thing we have more concentration of water. In one substance we have less concentration of water, but obviously which have the more concentration of water will have the more kinetic energy. The very best example will be the blood. So in our body, the muscle will have less amount of water in compared to the blood. So because of that, blood will have more kinetic energy, so it will be flowing thoroughly in our body. Or water potential. So blood is an example for this I am telling. So for example, if you take an example from the plant body itself, xylem. So xylem will makes the water to flow. So in that xylem you will possess a high level of kinetic energy because of water. Hence it is obviously that pure water will have the greatest water potential. Means more the water potential. So water concentration will be directly proportional to water potential. If two systems containing water or in contact, random movement of water molecules, means here and there movement of water molecules, will result in net movement of water molecules from the system with high energy to the one with the low energy. So, for example, minusing, it is like you have the two forces in an opposite direction. One force is of 10 newtons, one force is of 5 newtons. Now, the resultant force is 10 minus 5 newtons. So, likewise, the net force is the resultant force of all the water molecules. So, potential from having the lower water potential. This process of movement of substances down a gradient of free energy, we call it as diffusion. So, downhill transport or diffusion means it is moving from higher concentration towards the lower concentration. So, water potential is denoted by Greek symbol psi. So, P is silent here, psi, this is the symbol, and it is expressed in pressure units such as pascals. So, pascals pressure. By convention, the water potential of pure water at standard temperatures, which is not under any pressure, is taken to be 0. So, the water potential will be 0. Without the pure water will have a standard temperature. So, the water will not possess any temperature, any potential. So, it will be 0. This is very important question under competitive exams. If some solute is dissolved in pure water, the solution have a fewer free water molecules and the concentration of water decreases, reducing its water potential. Hence, all solutions have a lower water potential than the pure water. Okay, so because it will have less amount of water molecules, because all the solute will be binding towards the water molecules. Because of that, the potential will decreases. So as we already seen, water concentration is directly proportional to water potential. Hence, all solutions have a lower water potential than the pure water. The magnitude of this lowering is due to dissolution of solute, that we call it as solute potential. So, or psi s. Psi s is always negative. So, solute potential will be always negative. The more the solute molecules, the lower, means more negative will be the psi s. So, for a solution at an atmospheric pressure, water potential is equal to the solute potential at an atmospheric pressure. Okay, this point is very important. Water potential is equal to solute potential, nothing but psi w is equal to psi s during atmospheric pressure. If the pressure greater than the atmospheric pressure is applied to the pure water or a solution, its water potential will increases, means nothing but the water concentration is going to increase. That is the reason. So, because obviously you always remind water concentration is directly proportional to water potential. If the water potential is increasing, means water molecules are also going to increase. It is equivalent to pumping water from one place to another place. Can you think of any system of our body, in our body, where pressure is built up? Pressure can build up in a plant system when plants enter a plant cell due to diffusion, causing a pressure build up against the cell wall. So this we call it as turgid. For example, inside the plant cell, the water is going to enter. So the limiting wall for a plant cell is cell wall. So the membrane will be keep on increasing in size. So it will be very tough, and this pressure we call it as turgor. Cell will be turgid, means very tight in nature. Like for example, you are blowing the balloon. You will be keep on blowing the balloon. At some extent, you will feel the tightness in the balloon. So the air pressure will be more increased. If you cross that limit, what will happen? The balloon will burst. So to certain limit only it can hold that very tight pressure, which we call it as turgid. This increases the pressure potential. So now the pressure potential will get increases. You assume the balloon with air. So the pressure potential is usually positive. So from here we came to know the water potential will be positive, solute potential will be negative, and the pressure potential is usually positive. Although in plants, negative potential or tension in the water column in the xylem plays a major role in water transport up to the stem. For this, the very best example is, for example, we are drinking a cool drink with the straw. So we are creating a negative pressure. Because of the negative pressure only, the cool drink or a drink is coming into our mouth. If we are creating a positive pressure into that straw, you can see the bubbles. So the same thing will be happening here also. Inside the xylem, the negative pressure has to be created. Then only the water will move from the roots towards the stem. Pressure potential is denoted by psi p. So water potential means psi w. So the same alphabets they're used. Solute potential means psi s. Pressure potential means psi p. Water potential of the cell is affected by both solute and pressure potential. So the relationship between them is as follows. Psi W is equal to psi S plus psi P. Means nothing but water potential is equal to, it is a sum of solute potential plus pressure potential. So this is very very important, and always the solute potential will be negative. If the solute potential is been increasing, means more getting more negative. So in an atmospheric pressure, the water potential will equal to solute potential. So that is also very important point. So for a pure water, the water potential will be 0. First, let's see about osmosis. What is osmosis? Osmosis is very easy, that if you remind the diffusion, what we are talking in diffusion, simple diffusion, it will be movement of molecules or solute or solvent, whatever the thing, movement from higher concentration to lower concentration. Mostly the best examples will be liquids and gases. But the same movement of water from higher concentration to lower concentration, that we call it as osmosis. Yes, we are talking here, the diffusion of water is nothing but osmosis. This is a very simple concept. If you understand about diffusion, the osmosis will be very very easy. The plant cell is surrounded by a cell membrane and a cell wall, because cell wall is a characteristic feature of plant cell. The cell wall is freely permeable to water. To understand this concept, you remind the cotton. Cotton is nothing but a cell wall. So you will have that cellulose. If you dip into the water, what will happen? The cotton. So it will absorb the water very fastly. Means it is freely permeable to water. And substances in solution, hence is not barrier to a movement. So cell wall is not a barrier. In plants, the cells are usually contains large vacuole. This is also a characteristic feature of the plant cell, whose contents, so inside this central vacuole we will have cell sap, vascular sap, we also call it as cell sap, contribute to the solute potential of the cell. Okay, so whatever the solutes, it will be present inside this vacuole. In plant cell, the cell membrane and the membrane of the vacuole, which we call it as tonoplast, the membrane of the vacuole we call it as tonoplast. So the tonoplasts together are important determinants of the movement in and out of the cell, because the major portion of the plant cell is occupied by central vacuole. Whatever the solutes, unwanted solutes, will be stored inside the vacuole. So that vacuole and the will create the movement of solutes or solvents. So whatever is needed, it will enter, make the cell will absorb. Whatever is not needed, the cell will not enter, allow it to enter at all. So let's see osmosis once again. So now we came to know, so the tonoplast, nothing but the vacuole, central vacuole is very important for movement of the particles in and out of the cell. Let's see about osmosis in detail. Osmosis is the term used to refer specifically to diffusion of water. As I already told you, it is very very simple. So the terminology will makes us a quite complicated thing. So in very simple terms, the diffusion of water is nothing but osmosis across a differentially or selectively permeable membrane. Always the biological membranes are selectively permeable, because if the biological membranes are freely permeable, what will happen? The water can come freely inside, it will increase the size and it will burst. So because of that, it is being made as selectively permeable. Osmosis occurs spontaneously in response to a driving force. The net direction or a rate of osmosis depends on both pressure gradient and concentration gradient. Pressure gradient is very easy. Always, for example, inside the balloon, the same example, the balloon is filled with more pressured air. So the pressure of the air inside the balloon will be very more when compared to the atmosphere. So always the air want to burst from the balloon and it has to go outside the environment. So it is moving from higher concentration to higher pressure gradient to lower pressure. So the same thing with the concentration gradient also. So you will have a very best example here. Water will move from a region of higher chemical potential to its region of lower chemical potential. So it is very simple. The water will move from higher chemical potential means hypotonic to hypertonic. So yeah, that is very simple here. The water will move from hypotonic solution to hypertonic solution. Let's see how it is. Of its region of lower chemical potential until equilibrium is reached. So, at equilibrium, the two chambers should have nearly the same water potential. So, we already discussed water potential is equal to solute potential plus pressure potential, right? Psi S plus psi P. You may have made a potato osmometer in your earlier classes. I think you all remind this stage in the school. If the tuber is placed in the water enters the cavity in the potato tuber containing a concentrated sugar solution due to osmosis. So here is the picture. We are going to discuss about this picture. So in which we have the two chambers. In this picture we have the two chambers, chamber A and chamber B, containing a solution separated by a semi-permeable membrane. Let's see about solution A, in which chamber we have a lower water potential. So here in the chamber B, the second point is solution in which you have a lower solute potential. So solution of which chamber will have a it is a question, sorry, once again it is a question they are asking. Solution in which chamber we have a lower water potential, solution in which chamber we have a lower solute potential. Okay, let's answer these questions line by line. First question is solution in which chamber you have a lower water potential. What we already discussed priorly, water potential will be directly proportional to water concentration, in which we have lesser concentration of water in chamber B. So, in chamber B we have lower water potential. Yes, for the first A, it is answer is B chamber. Next, solution in which you have lower solute potential, that is chamber A. In the chamber A, you will have lower solute potential. So here you have a lower concentration of solute. In which direction will the osmosis occurs? So the osmosis will occurs from chamber A to chamber B. So A to B. That is the answer. Which solution have a higher solute potential? I think you may guess that is chamber B. From the chamber, in the chamber B only you will have higher solute potential because it has higher solute concentration. Solute concentration is also directly proportional to solute potential. So obviously answer is B chamber. At equilibrium, which chamber will have lower water potential? So both will have an equal water potential. That will be maintained. If chamber 1 has psi of 2000 kPa and other has 1000 kPa, which is the chamber that have higher psi? So what will be the direction? Here they have given some of the problems. So these two we will discuss in the next phase. Let's continue the discussion. Let us discuss another experiment where a solution of sucrose in water is taken in a funnel. So here a demonstration of osmosis. Here they have taken a funnel in which sucrose solution. So taken in a funnel where a solution of sucrose is taken in a funnel is separated from pure water in a beaker by semi-permeable membrane. So the center funnel we have the sucrose solution and outside we have the pure water, and it is being separated by a semi-permeable membrane. You can see it is tied near the funnel and beaker. So you can get this kind of membrane in an egg. So if you break the egg, you will find a very thin membrane. That membrane is nothing but biological membrane. It is semi-permeable in nature. Remove the yolk and albumin through a small hole at one end of the egg and place the shell in a dilute solution of hydrochloric acid for a few hours. The egg shell will dissolve, leaving the membrane intact. So the water will move into the funnel, resulting in rise of the solution in the funnel. Okay. So rise of the solution in the funnel. This will continue till the equilibrium is reached. In case sucrose does diffuse out through the membrane, will this equilibrium will ever be reached? So because of that, so if you can see from this picture, the water is going inside this funnel to maintain the equilibrium. So this a Thurstall funnel is filled with sucrose solution and kept inverted in a beaker containing the water. So the water will diffuse across the membrane as shown in the arrows to rise the level of solution in the funnel. So this is of case A. In the case B, pressure cannot be applied as shown to stop the water movement in the funnel. So we can stop this water movement by applying the pressure. You can see the pistol. Some type of pistol is being applied here so that the water movement will not be happen. So external pressure can be applied from the upper part of the funnel such that no water will diffuse into the funnel through the membrane. So this pressure required to prevent the water from diffusion is in fact the osmotic pressure, and this is the function of the solute concentration. More the solute concentration, greater will be the pressure required to prevent the water from diffusing in. So greater the amount of solute, greater the pressure is needed to prevent the water from coming. Numerically, osmotic pressure is equivalent to osmotic potential, but the sign is positive. But the sign is opposite. Osmotic pressure is the positive pressure applied, while the osmotic potential is negative. We have to remind this. Osmotic pressure will be positive, but the osmotic potential will be negative. Let's see about plasmolysis. What is plasmolysis? The behavior of the plant cell or tissue with regard to the water movement depends on the surrounding solution. If the external solution balances the osmotic pressure of the cytoplasm, then it's said to be isotonic. So if the external solution is more dilute than the cytoplasm, it is hypotonic. If the external solution is more concentrated, then it is hypertonic. So this is very easy that if the external solution balances the osmotic pressure of the cytoplasm, right? So what is isotonic means? Having equal amount of solute and equal amount of solvent. We call it as isotonic. Hypotonic means less amount of solute and more amount of solvent. That is hypotonic. Next, hypertonic. More amount of solute and less amount of solvent. So this is very easy that you have taken three solutions. In one solution, you are putting more amount of salt. So the water is too salty. Then what we will think? So salt is more than the water. So then that type of solution we call it as hypertonic solution. So the terminology is based on the solute, not based on the solvent. In other solution, you have taken more amount of water and less amount of salt. So if you taste it, you will feel the water will be less salty. Then we call that type of solution as hypotonic solution. You have taken one more solution that you will have an equivalent between the solute and solvent. Then we call that type of solution as isotonic solution. So plasmolysis occurs when water moves out of the cell and the cell membrane of the plants shrinks away from the cell wall. Like it will be opposite to turgid, opposite to turgor. So this shrink, this occurs when the cell is placed in a solution that is hypertonic, has more solutes to the protoplasm. So this stage, so this first picture we call it as plasmolyzed, and the second picture is flaccid, and the third picture is turgid. So in the first picture you can observe the water is moved away from the cell into the external solution. So the plant cell is getting shrinked. So it is moving away from the cell wall. Here you can see some amount of water is being moved away from the cell. So the plant cell is becoming loose. In the third picture, the water is getting inside the cell. The water is getting inside the cell so that the cell is becoming increasing in size. So it will be turgor. So that we call it as turgor. So is placed in the solution that is hypertonic. So coming to the first picture, so this plasmolyzed cell, in this is the state because it occurs when the cell is placed in a when the cell is placed in a hypertonic solution, the water inside the cell is moving outside the external solution. So now it is getting shrinked. So the water moving out, it is the first lost from cytoplasm and then from the vacuole. The water, when drawn out of the cell through the diffusion into the extracellular fluid, causes the protoplast to shrink away from the walls. So the cell is said to be plasmolyzed. Means death of the plasmolyzed. The movement of water occurred across the membrane, moving from an area of higher water potential to an area of lower water potential outside the cell. So which occupies the space between the cell wall and the shrunken protoplasm. Protoplast is a plasmolyzed cell. If they have much space between the cell wall and the cell membrane, then we call it as plasmolyzed cell. Next, when the cell is placed in an isotonic solution, there is no net flow of water towards outside and inside. If the external solution balances the osmotic pressure of a cytoplasm, is said to be isotonic. When the water flows into the cell and out the cell are in equilibrium, then the cell is said to be flaccid, means a bit loose. So when the cell is placed in an isotonic solution, then the state of the cell will be flaccid. The process of plasmolyzed is usually reversible. When the cells are placed in a hypotonic solution, higher water potential or dilute solution, as compared to the cytoplasm, the water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall. This we call it as turgor pressure. So the pressure exerted by the protoplast due to the entry of water against the rigid wall, which we call it as pressure potential. So because of the rigidity of the cell wall, the cell does not rupture. So this turgor pressure is ultimately responsible for enlargement and extension growth of the cell. So what would be the psi p of placid cell? Which organisms other than the plants will process the cell wall? Okay, let's see the answers for these questions in the next video. Okay, my dear friends, I hope you understood the concept of hypertonic, hypotonic and isotonic. Okay, so if you like that video, please do like it and share it to your friends and comment the feedback if any doubts or any queries or any changes. Okay, my dear friends, thank you very much.